Now let us proceed with the discussion of what happens in the Meiosis I. Meiosis I again has these four phases, which are the PMAT1, Prophase I, Metaphase I, Anaphase I, and Telophase I. And these four happen in sequence. Now, for Prophase I has five substages, which are the leptotene, zygotene, pachytene, diplotene, and diakinesis, which again happen in sequence. In leptotene, the chromosomes start to become more compact, or the chromatin strands start to condense to become chromosomes. For the purpose of understanding the process more, notice that in the example figure, we have four chromosomes. Next is the zygotene. In zygotene, homologous chromosomes pair up, and in this process, the pairing up is called as the synapsis. Homologous chromosomes are the chromosomes which are almost the same in size, the same and the same in genes. Upon synapsis, the homologous chromosomes will be linked via a protein complex called as the synaptonemal complex. When this happens, together the two linked chromosomes form a bivalent or a tetrad. Notice that in the figure, the cell still has four chromosomes. After zygotene comes next is the pachytene. In pachytene, crossing over takes place, or the exchange of genetic materials takes place between the non-sister chromatids of the homologous chromosomes. This begins when certain proteins break apart the DNA of the two non-sister chromatids at exactly the same location. Each broken end of DNA is joined to the corresponding section of the DNA from the other chromatid, which is the non-sister chromatid. This is made possible by the enzyme called as recombinase. The area of the chromosome where crossing over occurs is called as the chiasma, or plural, chiasmata, and there would be one instance of these per chromosome, resulting in what we call as recombinant chromosomes since they now have recombined genes. At the end of this, in the figure, the cell still has four chromosomes. So after crossing over, the chromosomes would look like this, since they now have fragments coming from the other chromosomes, specifically the non-sister chromatids. Now in diplotene, diplotene is recognized by the dissolution of the synaptonemal complex and the tendency of the recombined homologous chromosomes or the bivalents to separate from each other except at the sites of crossovers or the chiasmata. At the end of this, in the figure, the cell still has these four chromosomes. The final stage of meiotic prophase I is diakinesis. This is marked by the terminalization of the chiasmata. During this phase, the chromosomes are fully condensed, and the meiotic spindle or the meiotic spindle fibers are being assembled to prepare the homologous chromosomes for the separation. By the end of diakinesis, the nucleolus disappears. Diakinesis also presents transition to metaphase, which brings me now to Metaphase I. In Metaphase I, bivalents align on the equatorial plate. Notice that unlike metaphase of mitosis, which has only one pile of chromosomes in the equatorial plate, in Metaphase I of meiosis, there are two piles of chromosomes which line up at the equatorial plate. The lining up of the bivalents in the equatorial plate happens in random fashion. The number of possible arrangement of these chromosomes in the equatorial plate can be determined using the formula 2 raised to the power of n, wherein 2 is constant and then the n is a number of chromosomes in a haploid set of a particular organism. So let us say, for example, that the dividing cell is a human spermatogonium, which means it has 46 chromosomes. If the cell is in Metaphase I, how many possible arrangements of the chromosomes will there be? Again, use the formula 2 raised to the power of n. The answer for this one, wherein n, by the way, is a number of chromosomes in the haploid set. Therefore, from 46, the value for n is 23. Therefore, 2 raised to the power of 23, that's 8,388,608 possible arrangements of chromosomes in Metaphase I. In Anaphase I, the homologous chromosomes separate and are pulled toward the opposite poles by the spindle fibers. In the separation of the homologous chromosomes, notice that the sister chromatids remain associated with each other at their centromeres, unlike in the mitosis. Lastly, in the telophase, the nuclear membrane reappears or reforms. At the end of meiosis I, there are now two nuclei. Notice now that from 4 chromosomes in the earlier phases, we will now be having 2 chromosomes per nucleus. After telophase comes next would be cytokinesis, and we get 2 haploid daughter cells because they now only have half of the number of chromosomes of the parent cell, which was also evident in the figures shown, wherein in the first phases we had 4 chromosomes in a cell, and then in the produced cells they only have 2. In the 2 daughter cells produced, they only had 2 chromosomes each. These daughter cells are haploid because they only contain half the total number of chromosomes of every cell of an organism. So for us, for humans, again, if the dividing cell is the human spermatogonium, from 46 chromosomes at the end of cytokinesis after meiosis I, each of the 2 daughter cells will only get half of it, having 23 chromosomes each. Hence, they are called as haploid. In general, this is what happens in meiosis I and cytokinesis. So in Prophase I, Metaphase I, Anaphase I, Telophase I, and then cytokinesis. For some cells, they enter interkinesis, which is the period of rest that cells of some species enter during meiosis. This happens between meiosis I and meiosis II.